Hi, AP Biologist. This is Mr. Wolf, and today we're going to be talking about eukaryotic gene regulation. This is Objective 6.5 and 6.6 from the AP Biology curriculum. So the big question here is how are our genes turned on and turned off in eukaryotes? We previously talked about prokaryotic gene expression, which is much simpler, but eukaryotes have to deal with a lot more cells and a lot more specialization. So the question is, how do cells with the same genes, since we have all the same genes in all of our cells, differentiate to perform complex and very different specialized functions? As you can see here on the upper right, we have a sperm cell fertilizing egg cell, which resulted in a zygote, which was our original package of DNA. And through development, we've developed specialized structures and features all through the complex variations on how genes control or how genes are turned on and turned off, essentially. So in talking about evolution of gene regulation in prokaryotes, which are single-celled organisms, they have evolved to grow and divide rapidly. They have to respond to an ever-changing environment because their external environment is constantly fluctuating, and this requires them to exploit transient resources that are not going to be available all the time. So therefore, their gene regulation has to be able to be very quick and flexible and reversible. So certain genes need to be turned on and turned off rapidly depending on what's available. Kind of like we talked about the lactoperon, that gene can be turned on when lactose is available, making those enzymes available to digest that lactose, and then turn that gene off as to not waste resources when lactose is not available. They also have to be able to adjust enzymes for synthesis and digestion, just like we talked about for lactose, and then the tripoperon for synthesis. Prokaryotes need to be able to quickly change their gene expression and use resources when they're available. Now, for eukaryotes, since we are multicellular, our cells take on different functions throughout our lifetime. We've evolved to maintain a constant internal environment, despite what's changing externally. We maintain constant internal conditions, maintaining homeostasis, so gene regulation plays a big part in that. We have to regulate our body as a whole. We have to regulate growth and development. So it's a very long-term process, turning certain genes on for growth and then turning them off when we want to stop growing. Same thing for development of certain organs. And then these organs are specialized tissues, or made up of specialized tissues that have specialized cells. And we need to be able to turn on and off large numbers of genes to make those cells do certain things while not wasting resources and producing unnecessary proteins. And then as a whole body, we have to coordinate as a whole, rather than just serving the needs of one cell, all of our cells are kind of serving the needs of the body, and that takes a lot of communication and regulation in terms of our genome. So there are several points of control in eukaryotic organisms. A lot of the stuff we talked about for prokaryotes was all pre-transcription. So once the DNA was transcribed into RNA, protein synthesis was pretty much going to happen. So prokaryotes need to prevent mRNA from being made as a way of controlling if proteins are going to be made. Now, in eukaryotes, there's a lot more different steps or places where gene regulation can occur. We'll talk about all 7 of these as we go through, but one of the biggest reasons we're able to control or have different points of control is the fact that we have a nucleus where the mRNA is not able to leave until it's been processed. So we prevent translation from happening until all this stuff that goes on the nucleus happens. So starting off with the DNA packing. So basically we've already talked about this a little bit this year, that DNA is incredibly long in terms of how much DNA is found in our cells, and we have to basically pack it up and fit it in a very tiny space. And so the DNA goes through several different packing kind of stages or levels per se, where you have the double helix, which then spins around these histones, which make up these nucleosomes, and then those nucleosomes kind of bundle together to make these chromatin fibers, and those chromatin fibers kind of start to loop, making these loop domains, and then eventually we get to our big old chromosome. Just a reminder, we only see those chromosomes during cell division. Normally, our DNA is in the chromatin state when it's being used to actually build proteins. So a nucleosome, I want you to think about nucleosomes are kind of like beads on a string. It's our first level of DNA packing, and basically you are wrapping the DNA molecule itself around these histone proteins, which are actually 8 protein molecules all bundled together. They have a positive charge. Since DNA is negatively charged, it binds very tightly to those histone proteins and give us kind of a packing mechanism of how DNA is going to be organized. So now that we've talked about kind of how DNA is packaged up, this is where our first level of pre-transcriptional control actually comes into play. Most eukaryotic gene regulation comes before transcription, very similar to prokaryotes. Conserves energy, conserves resources. So there are two main pre-transcriptional control mechanisms. We have what are called epigenetic tags. Maybe you've heard of epigenetics before. And then we have transcription factors, very similar to prokaryotes in that they're specialized proteins that help recruit the RNA polymerase for transcription to actually happen. So epigenetic tags, there are degrees of packing in DNA that regulates transcription. So again, that DNA is wrapped around histones. Sometimes they're tightly wrapped around those histones where there's no transcription is going to be taking place. Basically, if it's packed tightly, the RNA polymerase can't get to the actual genetic code and no transcription will actually be happening. Essentially, this is the genes being turned off. We call this heterochromatin. It's the more condensed regions we actually see in the nucleus, as you can see in the bottom right picture. We also have euchromatin, which are lighter regions of DNA, and that's where the DNA is kind of packed a little more loosely. This is the actual part of the chromatin that's being used by the cell for transcription. And so there are degrees in which we can pack that DNA up and turn some genes off and some genes on based on how tightly those histone proteins are kind of collecting together. Part of the process that controls this histone packing is called acetylation. So acetylation of histones basically helps unwind the DNA. So this loosely wraps around the histones, enables transcription to take place. Essentially, the genes are turned on. So basically what's happening is that acetyl groups are attached to the histones, and that makes the histone proteins change shape. And as we've been learning in AP biology, when something changes shape, it changes kind of what it does. And so when those histone proteins change, transcription factors now have an easier time getting to the genes, and RNA polymerase can now get to the genes and actually start transcription. So basically, the active chromatin, those histone proteins are like more spread out, whereas their inactive chromatin, they're more condensed and clustered together because they don't have those acetyl groups attached to them. So I want to talk about DNA methylation. Methylation of DNA blocks transcription factors from actually getting to the DNA. So no transcription takes place. Essentially, it's turning the genes off. Methylation is where we attach a methyl group, a carbon and 3 hydrogen group, to the cytosines, and that permanently inactivates the genes. Well, most of the time. There's been some studies showing that this could be reversed in some cases, but most of the time this creates a permanent inactivation. One good example of this is how many of the chromosomes or how the bar bodies are created during inactivation of the mammalian X chromosome. Basically, all the cytosines get these methyl groups that don't allow the RNA polymerase to actually transcribe the genes. There's a really cool simulation here at learn.genetics.utah that shows this acetylation and methyl tags being added, allowing the histone configuration to change. So basically right here, we see the DNA molecule itself, and it's wrapped around those histones. You see down here how much of this GFP mRNA is being able to be produced through this acetylation and methylation. And then you see how much protein is being produced, and the cell's going to glow more green the more protein that's being made. So as I turn this knob, you can see that there are more acetyl groups attaching. You can kind of see them attaching to the histones, and those histones are being pulled apart, and the methyl tags are coming off. Oops, went too far. Coming off the DNA. Or as you can see, there's a lot more mRNA being made, a lot more green fluorescent protein being made. The cell is bright green. And then as I turn this wheel the other way, the methyl groups are being added, the acetyl groups are coming off the histones, and that DNA is being packed up and compacted more into heterochromatin, and the mRNA's levels are going down, green protein is going down, cells not glowing green. Basically, we get to a point where we can't access those genes anymore because DNA is all packed up. So that's a great example of how you have heterochromatin, DNA is all packed up, the histones are not acetylated, and then you start to acetylate them and the DNA gets stretched back out. We can actually access the genes, start transcription, and then go through translation. And so this kind of gets into epigenetics, where epi kind of means above. So it's kind of like how your genes are controlled beyond just what they are. Your genes don't really change throughout your lifetime, but the expression of them changes. You are born with a certain set of genes, but how those genes are turned on and turned off can be influenced by environmental factors. Helps explain a lot of things, differences between identical twins, how certain cancers form over time, how certain carcinogens can affect our genome. And what's interesting is a lot of research is being done, and there is studies that show that even changes in your epigenome can be passed on to your offspring. So your diet essentially could affect what genes are turned on and turned off in your offspring, for example. So how do we initiate transcription? So basically, we have these control regions on our DNA. They're called promoters, and those promoters are nearby the actual gene sequence, and they control where the RNA polymerase is going to bind. And this is kind of signaled by these transcription factors that find the promoter. This kind of gives you a base rate of transcription, so gives you kind of like what would be considered a baseline transcription level. And then enhancer regions actually can help promote or increase mRNA synthesis or increase transcription. So these enhancers are distantly controlled sequences of DNA that bind to what are called activator proteins. And those activator proteins help enhance the rate of transcription. Essentially, you get more transcription, you get more translation, you get more protein product made. So enhancer DNA sequences, again, are distant controlled sequences. They're controlled by activator proteins, and those proteins bind to the enhancer region, the enhancer sequence of the DNA. So it could be hundreds or thousands of base pairs up the DNA sequence, or before the gene, I should say, upstream of the DNA. And then those activators kind of bend that DNA over and allow those transcription factors to bind and therefore increase transcription, again, increasing protein production. There are silencer proteins that do bind to those enhancer regions, and they basically block those transcription factors from collecting and can prevent the gene from being read. So activators can bind close to or far away from the gene of those enhancer regions. When enhancers are far away, DNA actually can bend and activate the general transcription factors together. And then very similar to prokaryotic gene expression, we have repressor proteins that will bind to that promoter region and block the transcription factors, preventing from RNA polymerase from ever attaching to the actual DNA molecule itself. If you're blocking transcription, you get no translation, you get no protein. Here's just another example, another picture of it. So again, those activator proteins are binding to enhancer regions. Some genes require several enhancers to be activated to turn on. You've seen this in the sickle cell fish example that you just did. And then those enhancer sites are usually hundreds or thousands of bases upstream of the gene. And then the transcription factors bind at the promoter, usually has a TA type sequence to it. And that's part of the initiation complex, where that promoter is going to have a collection of transcription factors. It kind of gets the RNA polymerase in the right spot. And when all those things connect and combined, then RNA polymerase is able to read the gene and transcription will occur. There are different activators and repressors present in different cell types, so different genes can be turned on. So you see this example here. In a liver cell, you have a certain set of enhancer regions in front of this gene. You have the right available activators, and this albumin gene will be expressed, whereas it doesn't have the right activators to express the crystalline gene, which will be expressed only in the lens cell of your eyes. So these lens cell nuclei will produce the crystalline gene product, whereas they will not have the right activators to produce the albumin gene product that is found in the liver cell. So it's what makes the liver cell turning certain genes on and the lens cells turning other genes on based on the activator proteins that they contain. So transcription factors play a very important role in determining what cell type the cells will become. So when stem cells start to specialize. Certain transcription factor genes are turned on and turned off. This is all done through a series of chemical messages that are received by those stem cells, and that allows for differentiation to occur and cell specialization to occur to do certain jobs in our bodies. All right, now we're going to move into post-transcriptional control. In post-transcriptional control, transcription has occurred. Now we control what is going to happen with the mRNA. We've kind of already talked about this earlier when we talked about transcription. You have that alternative RNA splicing that can occur, where you get variable processing of exons that creates a family of proteins. So certain exons that are spliced together will create different combinations of mRNAs, which will eventually lead to different proteins being produced. Now, these proteins will probably be very similar because they have similar exons as part of them, but it explains how one gene now can actually code for multiple proteins. We also have the lifespan of mRNA can be determined by the amount of, or how long that poly A tail is in that 5' cap. Basically, the longer the mRNA lasts in your cells, the more protein product it can make. So depending on how much protein the cell wants to make, it can last from several hours to several weeks, depending on how long that poly A tail is and what kind of 5' cap is added. Another thing that is controlled or can control your mRNA Another thing that can control another type of post-transcriptional control is microRNAs and small interfering RNAs. MicroRNAs, or miRNAs, are small single-stranded RNA molecules that bind to the messenger RNA, and basically they kind of mark that RNA for degradation, or they can block translation. Essentially, they can kind of silence the mRNA. Even though the mRNA was made, these microRNAs have control over if that mRNA is going to be read and translated or not. It is estimated now that the expression of at least half of all human genes are regulated by miRNAs. There's a lot more research going into these, and it's kind of the newer field in terms of our understanding of how genes are turned on and turned off. Sometimes it's after transcriptions actually happened. A lot of that's done by what are called small interfering RNAs. siRNAs are similar to microRNAs in their size and their function, but they block the gene expression by using what's called RNA interference, or RNAi. They've actually started using this in laboratories as a means of disabling genes and investigating their function. Essentially, they can turn specific genes off and see how cells in the lab respond to that one specific gene, and getting more of an insight as to what that gene and what that protein actually does for the cell. So here's how those microRNAs kind of work. They work within a microRNA protein complex, and they are essentially binding to certain target mRNA sequences, and either mark that sequence or that mRNA to be degraded or broken down, degraded, or they can help block translation. Either way, if the microRNA is complementary to the mRNA, no translation is going to take place, no protein is going to be produced. Finally, we can control translation too. We can block the initiation of the translation stage. There are regulatory proteins that attach to the 5' end of the mRNA, and that's where the small subunit of the ribosome attaches, and that prevents the attachment of the larger subunit and that initiator tRNA, that tRNA that's carrying methionine, essentially blocking translation. So there are regulatory proteins that will prevent the protein from being produced at the ribosomal stage. And finally, we also have ways of breaking down proteins. Once proteins are produced, they can be processed and essentially gotten rid of when they're done doing their job. So protein processing involves folding, cleaving, adding sugar groups, targeting for certain transports, basically modifying the protein post-production. It's kind of what the Golgi apparatus and the ER are involved in kind of sending proteins on their way to do their jobs, whether it's in the cell or somewhere else as like a hormone or a signaling molecule that's being sent outside the cell. And then you have protein degradation, where proteins eventually get broken down. Everything in a cell is going to be recycled. And so ubiquitin is a chemical that attaches to essentially proteins that the cell is marking as this needs to be broken down. It kind of marks this is the protein that we need to break apart and reuse its pieces. This was discovered in the 1980s, and these three gentlemen won a Nobel Prize in 2004 for discovering that ubiquitin was kind of like this death tag. It marked unwanted proteins with a label, and then these proteins would be broken down by these proteasomes, which are kind of like garbage disposals for proteins found in your cells. And so cells that no longer want to use those proteins mark them with ubiquitin, and they get kind of chopped up, and those pieces get reused to make new proteins that the cell actually needs. Here's kind of what a proteasome looks like. It's a protein degrading machine. It's kind of like a cell waste disposer. Again, it breaks those proteins down into small amino acid chunks, which is basically useful for cellular recycling and reusing those amino acids to make new proteins. So that is a lot of stuff that we've talked about. So gene regulation, from the very beginning, you have transcriptional control, where you have the packing of the DNA. You have transcription factors that mark which genes are going to be turned on and turned off. You have post-transcriptional control, where you have mRNA processing. You have splicing of certain exons together. You have that 5' cap and the poly A tail that gives the mRNA kind of a lifespan. And then those mRNAs can be broken down or basically stopped by siRNAs. You have translational control, where you can block translation using kind of repressor proteins. And then you have post-translation control, where you process proteins to be exported or sent to different places in the cell by adding different groups to it. And you go through protein degradation when those proteins are no longer needed by the cell by marking them with ubiquitin and kind of sending them through the shredder to be reused to make new proteins. So there's your initiation of transcription, kind of getting the process started. Again, that's all involved with what DNA is unpacked and what transcription factors and activators are available. There you have your RNA splicing. All this takes place in the nucleus. So before it leaves, you have these levels of control. Once mRNA leaves, you can protect it, give it a lifespan by how long the poly A tail is and what that 5' cap is doing. You then process it. Initiation of translation can be blocked. And then you have your protein processing and protein degradation. So there you have it, all the ways eukaryotic organisms can control protein production. So I hope this was helpful, and thanks for watching.